A very good morning to all of you. Today we are talking about turgidity, flaccidity, and plasmolysis. Turgidity and flaccidity. Yes, what is turgidity and flaccidity? Turgid plant, flaccid plant. Suppose you are going for a trip for 1 week. After you came back, yes, your plants become dried or wilted, or all the leaves are in drooping condition because of the scarcity of water. Because these plants are not getting enough water, then the cells of the plants are not getting enough water. That condition we call it as flaccid. Then this is flaccid plant. Suppose you are watering every day, the plants become fresh, right? Then all the cells of the plants are getting enough water. All the cells are in turgid condition. That type of plant we call it a turgid plant. We are discussing turgidity and flaccidity in detail in the coming slides. Every living plant cell, directly or indirectly, is in contact with fluids. The root hairs are surrounded by soil water. All such cells are subjected to osmosis. These root hairs are unicellular. You know that in a single root, enormous number of root hairs are there. Then the water concentration or water potential is more in the soil water compared to the cell sap of root hair. Then cell sap contain high solute concentration, less water concentration. Then water move from soil to the cell sap. That is osmosis, right? Through a semi-permeable membrane. Then what is the relation with osmosis and turgidity? Yes, we are discussing that. How can we relate turgidity and osmosis? First, we can see what is turgidity. When a cell reaches a stage where it cannot accommodate any more water, that is, it is fully distended, it is called turgid. The cell is turgid. We can say, yes, the cell cannot accommodate any more water, or cell is in distended condition. That is called turgid. The cell is turgid. We can say the cell is turgid. Then that condition is called as turgidity. Then, when this turgidity will occur? When a cell is placed in hypotonic solution. When a cell is placed in hypotonic solution, endosmosis will occur. Water move from outside to inside because in hypotonic solution, water concentration is more, solute concentration is less. That's why water will move from outside to inside. That is endosmosis. Then, when a cell is turgid means which type of osmosis will occur here? Endosmosis will occur. Turgidity. What is the definition of turgidity? Turgidity is a state of the cell in which the cell wall is rigid and stretched by an increase in the volume of vacuoles. Cell wall is stretched by an increase in the volume of vacuoles due to the absorption of water. Here endosmosis will occur. Yes, due to endosmosis, the cell becomes turgid. Then that condition is called turgidity. What is Turgor pressure and wall pressure? When a cell is turgid, this is a turgid cell, its wall is stretched under pressure. The cell wall is stretched under pressure from inside. Here you can see inside the cell, what is there? Cell contents are there. Then wall is stretched under the pressure from inside. It presses the cell contents towards the center of the cell. Then this cell wall is also applying a pressure towards the cell content. Here two types of pressure we can see, Turgor pressure and wall pressure. What is Turgor pressure? The pressure of the cell contents. The pressure of the cell contents. You can see is here inside the cell is cytoplasm, cell organelles, right? Then cell sap. These all the substance inside the cell, that is cell contents. These cell contents, when the cell is turgid, the cell contents applying a pressure on the cell wall. Here you can see on the cell wall, that is called Turgor pressure. Who is applying the pressure on the cell wall? Cell contents applying a pressure on the cell wall. That is called Turgor pressure. Then what is wall pressure? It is just opposite to Turgor pressure. The pressure exerted by the cell wall. Cell wall is applying a pressure where on the cell contents. That is called wall pressure. Here a cell wall is applying a pressure on the cell contents. That is called wall pressure. Plasmolysis and flaccidity. A turgid plant cell kept in 5% salt solution. Suppose the cell is kept in 5% salt solution for a few minutes, a few hours. Then what will happen to that cell? It will lose its distended appearance. After half an hour or 1 hour or after a few minutes, this cell, turgid cell, will lose its distended appearance. Then here you can see the cytoplasm will shrink and the plasma membrane will withdraw from the cell wall. This is cell wall. Then yes, plasma membrane will withdraw. Very important. Yes, plasma membrane will withdraw from the cell wall and the cytoplasm will shrink. This shrinkage from the cell wall is called plasmolysis. This is the process, or we can say phenomenon, by which the cytoplasm will shrink and the plasma membrane withdraw from the cell wall. Here you can see that is called plasmolysis. Then the cells in this state, we call it as limp or flaccid cell. This cell we call it as limp cell or flaccid cell. Then that condition is called flaccidity. This condition we call it as flaccidity. Then when it will happen, this plasmolysis or flaccidity, is when a turgid cell placed in hypertonic solution, we can say, right? 5% salt solution means inside the cell, solute concentration is less compared to outside. Then when a cell is placed in hypertonic solution, plasmolysis will occur. Then what type of osmosis is happening here? Then actually how it will shrink, this cytoplasm will shrink or plasma membrane will withdraw, because the water molecules present inside the cell contents or inside the cell will move out of the cell, because outside the cell, what is there? Hypertonic solution is there. Then there water concentration is very less. Outside water concentration is very less. Inside this water concentration is more, solute concentration is less. Then outside water concentration is less and solute concentration is more. That's why water molecules move from inside to outside. Then what type of osmosis is happening here? Yes, during plasmolysis, very important, exosmosis will occur because cell is placed in hypertonic solution. We can go through some definitions. First one, plasmolysis. Plasmolysis is the contraction of cytoplasm and withdrawal of plasma membrane from the cell wall, caused due to the withdrawal of water when the cell is placed in hypertonic solution. Here you can see this is a normal cell, turgid cell is placed in hypertonic solution. Cytoplasm will shrink and plasma membrane will withdraw from the cell wall. Here you can see caused due to exosmosis or caused due to withdrawal of water from the cell to outside when the cell is placed in hypertonic solution. Very important. The cell is placed in hypertonic solution. This is the definition of plasmolysis. Then what is deplasmolysis? A plasmolysed or flaccid cell. Here you can see. This is just opposite to is plasmolysis. Deplasmolysis is the opposite of plasmolysis. A plasmolysed or flaccid cell, this is a flaccid cell, is placed in hypotonic solution or it will return to the water before it is dead. Before it is dead, its protoplasm again swells up, pressing tight against the cell wall. Here, when a flaccid cell is placed in hypotonic solution before it is dead, what type of osmosis will occur? Endosmosis will occur. Then cell again is turgid. Then the recovery of or reversal of plasmolysis. Recovery or reversal of plasmolysis is called deplasmolysis. At the time of deplasmolysis, the cell again turgid. The flaccid cell again turgid. At the time of plasmolysis, turgid cell become flaccid. I hope you understood. Plasmolysis is happening is when a cell is placed in hypertonic solution. Exosmosis will occur here. At the time of deplasmolysis, is that plasmolysed cell or flaccid cell is placed in hypotonic solution. Then endosmosis will occur. Then cell become turgid. The next definition, that is flaccidity. Flaccidity is the condition in which the cell content is shrunken and the cell is no more tight. That condition we call it as flaccidity. The cell is no more tight. The cell is then said to be flaccid. We can say flaccidity is the reverse of turgidity. You already know what is turgidity. Yes. Then flaccidity is just opposite of turgidity. What are the uses of turgidity to plants? We are discussing one by one. First one, turgidity provides rigidity to soft tissues such as leaves. Here you can see in this plant, this is a turgid plant, leaves are rigid. Here all the cells of this plant are getting enough water, or we can say the cells are in distended condition. Cells are turgid. Then here you can see, observe the other plant. If you observe the other plant, this plant is in wilting condition. You can see is all the leaves are drooping. All the leaves are is in wilting condition. What is the reason for that? Wilting of leaves is usually noticed when a plant is exposed to hot afternoon sun. In the morning, if you are watering the plant, then yes, but the intensity of light is very high in the afternoon. That time transpiration rate will be more compared to water absorption rate. That time wilting will occur. More amount of water will escape from the plant to the atmosphere. That's why transpiration rate will be more compared to absorption rate. That's why wilting will occur. But in the evening, what you can observe, in the evening when the transpiration rate reduced, you know that intensity of light is less compared to afternoon, right? In the evening, the transpiration rate is reduced because intensity of light is very less. Then the quantity of water absorbed exceeds the loss of water through the transpiration. Then absorption rate will exceed the transpiration rate. That's why the turgidity of the leaf cells is restored and the leaves again stand out. In the evening, you can see these leaves again stand out or leaves become rigid again. Another use of turgidity, that is, turgor pressure helps to push through the hard ground. Examples: mushrooms, seedlings, and sometimes the roots of certain trees have been seen to crack the walls or a concrete floor of an adjoining building. What is the reason for that? This is due to turgor pressure. Then this portion is very important for give reason. Question will come like that. Mushrooms, seedlings, and sometimes the roots of certain trees have been seen to crack the walls. Give reason. Yes, this is due to turgor pressure. You can write the answer like this. Turgor pressure helps to push through the hard ground. Another point, turgor in root cells build up root pressure. Turgor in root cells build up root pressure. Here we can see one demonstration experiment. Here, if you cut a well-watered pot plant a few centimeters above the soil and immediately fix a glass tube by means of rubber connection, water will start coming out of the cut end. Here you can see this is a cell sap of water coming out of the cut end and rise up in the glass tubing. This rising water can raise the mercury filled in a connected manometer. Here is we can fix a manometer also. Then this rising water or cell sap can raise the mercury that filled in a manometer. What is the reason for that? Yes, this upward flow of water is due to heavy pressure from the roots, which is called root pressure. This can raise the mercury. Then what is the definition of root pressure? Root pressure is the pressure developed in the roots due to the continued inward movement of water through the cell-to-cell osmosis, which helps in the ascent of cell sap upward through the stem. That is called root pressure. Then root pressure will occur when there is continued, very important, continued inward movement of water through the cell-to-cell osmosis. That's why we are saying we are taking a well-watered pot plant. Then only this experiment will occur. Or we can observe the root pressure here. The cell sap will come out is only in well-watered plant. Another point you have to remember, that is loss of water through the cut stem is called bleeding. Loss of water or cell sap through a cut stem is called bleeding. Very important. And what is the reason for bleeding? Question may come. Then this is due to bleeding is due to root pressure. This is in the case of plants. Don't confuse with that. Yes, what is root pressure? We already discussed that in our earlier slide. The root pressure is built up due to cell-to-cell osmosis in the root tissues. Here you can see these are root tissues. Then root pressure is developed due to cell-to-cell osmosis in the root tissue. If you cut off the shoot of a plant, the water pushes out from the root stem. Yes, you can see here. This is due to root pressure. Then question may come like this. If you cut off the shoot of a plant, the water pushes out from the root stem. What is the reason? This is due to root pressure. Turgor in the opening and closing of stomata is the another use of turgidity to plants. Here you can see in this picture, this is open stoma, this is closed stoma. The opening and closing of stomata depend on the turgidity of the guard cell. Opening and closing of stomata depend on the turgidity of the guard cell. What is the difference between stomata and stoma? Here we can see is stoma, this is stomata. What is the difference? Stomata is the plural form, stoma is the singular form. Then if the guard cells, these two are guard cells, these guard cells are turgid, then stoma will open. If the guard cells are flaccid, stoma will close. Turgor movement is the another use of turgidity to plants. Examples, the rapid drooping of the leaves of sensitive plant. Mimosa pudica is a sensitive plant. The common name of Mimosa pudica, that is touch me not plant. If we touch the leaves of this plant, what will happen? It will droop very fastly, right? Yes, what is happening here? In this plant, the stimulus of touch leads to loss of turgor. Very important, loss of turgor at the base of the leaflets. Where? At the base of the leaflet and the base of the petioles. Here you can see. This is the petiole. What is petiole? Stalk of the leaf. Yes. Then base of the petiole, the swollen region. That swollen region is called pulvinus. Then the stimulus of the touch leads to loss of turgor at the base of the petiole and at the base of the leaflet. You can see the pulvinus clearly here. Yes, this is the stalk of the leaf. Then the base of the petiole is swollen. That region we call it as pulvinus. The next point, insectivorous plant leaves close up to trap prey. Here you can see this is an insectivorous plant. Which plant is this? This is pitcher plant. This is the leaf modification. Then insectivorous plant leaves close up to trap prey. This is due to Turgor movement. Bending movement of certain flowers towards the sun. Here you can see sunflower and the sleep movements of the leaves of certain plants at night. This is moringa leaves or drumstick leaves, right? Yes, the sleep movements of the leaves is all due to Turgor movement. What is the reason for this Turgor movement? Do you know imbibition and Turgor together generate much force? Imbibition is the passive absorption of water by substances such as cellulose, proteins, and starch. Turgor is the pressure setup inside the plant cells due to hydrostatic pressure on the cell walls on account of incoming water as a result of endosmosis. Example, seeds and grains swell up when soaked in water. Here you can see. Seeds and grains swell up when soaked in water is due to imbibition and Turgor. These both together generate much force. What is guttation? In certain plants like tomato, grass, banana or ferns, the root pressure is high enough to force the water all the way through the stem and comes out through the end of the leaf veins. That is called guttation. You can see water drops comes out through the veins, leaf veins due to root pressure. This process we call it as guttation. This you can see in tomato, grass, banana or ferns, usually in herbs. Then this guttation happens due to root pressure. These water droplets will come out through the leaf veins due to root pressure. That process is called guttation. Revision. Answer the following. First one, state of a cell when it cannot accommodate any more water. Turgid. Pressure of the cell content on the cell wall. Turgor pressure. The condition that is opposite to turgid. Flacid. The state of a plasmolyzed cell after the re-entry of water. The state of plasmolyzed cell after the re-entry of water. Answer is deplasmolyzed cell, or we can say turgid cell, right? Deplasmolyzed cell is a turgid cell. The pressure under which water passes from the living cells of a root into the xylem. Root pressure. Give reason. If you sprinkle some common salt on grass growing on a lawn, it is killed at that spot. Here we are sprinkling some common salt on grass. Then the solute concentration is more outside than inside the cells of the grass. Then water molecules move from the cell to outside. That's why all the cells of the grass become flaccid due to exosmosis, because water concentration is more inside the grass cell compared to outside. Then next statement, a plant cell when kept in hypertonic salt solution for about 30 minutes turns flaccid. Here, a plant cell we are keeping in hypertonic salt solution. Then water concentration or water potential is more inside the plant cell. Here also exosmosis will occur. Water molecules move from plant cell to the salt solution. That's why plant cell become flaccid. Potato cubes, when placed in water, become firm and increase in size. We are keeping potato cubes in water. Water is a hypotonic solution. Then water concentration or water potential is more outside. That's why water molecules move from outside to inside the potato cubes. Endosmosis will occur. Then that's why potato cubes become firm and increase in size. All the cells of the potato cubes become turgid. Define the following: turgidity, flaccidity, plasmolysis, and root pressure. These are important definitions. You have to learn this. Thank you, children. Have a good day. If you like my channel, please like, share, and subscribe. Thank you.